Hello everyone, and once again I welcome you all to Misostudy. So we were discussing about the chapter that is strategies in enhancement of food production. In strategies of enhancement of the food production, we have completed the zoology part and we will be doing the botany part. In the just previous class, I was talking about the plant breeding experiments, the different plant breeding strategies, how we can employ these plant breeding for the enhancement or for the production, for the more and more production of the food. Not only for the production of the food, but also they provide a different kind of resistance to the plant in terms of the pest, in terms of the bacteria, in terms of the mycoplasma. Another important function of this application of this plant breeding is that we can increase the yield, we can increase the plant variety as well, which is suitable for one particular type of environment. So we have discussed some general introduction regarding the plant breeding, and in this class I will be talking about what are the strategies, different strategies starting from the collection of germplasm to the different employment or to the formation of a new, you can say, the variety. So we will be discussing about the different steps which are involved in the plant breeding strategies that are employed by the government as well as non-government agencies. So let's start the topic. The topic is plant breeding. Plant breeding means we are mating two plant species, we are mating to those two varieties of the plant, and one is having one particular kind of characteristics which is suitable for the environment, whereas another variety is having that character which is suitable for their environment. Or suppose let's talk about one of the species that they can produce more and more amount of the proteins. Suppose the product of this particular plant is having more protein, and product of the same plant, same variety, or you can say the germplasm, or moreover I would rather say the genotype is same, but some of the characters they are different, and because of the change in this particular plant, we can see that they are resistant to the plant. So by breeding, by mating these two individuals, these two plants, I'll get that particular plant who will be having the dominant or the main characteristics which I am looking for from the parent 1 and from the parent 2. So how these different strategies they could be applied in terms of the plant breeding. Now in the previous class I was talking about the conventional breeding system. Conventional breeding system, that means a mating between the pure lines. I have, by the selfing experiments, by the homozygosity, increase in the homozygosity, I'll get the two parents, I'll cross those two parents, and after this what I'll get is I'll get the mixture of these two. Right, this was our conventional method of the breeding. With the introduction of the plant tissue culture, with the introduction of the biotechnology, and with the introduction of the molecular biology, a drastic change in the plant breeding system has been done. Because of these particular changes, we are now having those varieties which are resistant to the pest. We are having those varieties which are resistant to the bacteria, mycoplasma. We have those varieties which are resistant to the various environmental resistance such as drought, such as water scarcity, drought and water scarcity, they are same, and such as these are those plants which give a better yield, the more yield they give, and second, they are more in the nutritive quantity. So let's talk about the plant breeding and why we go for the plant breeding. Because of the various strategies which is followed with the help of plant tissue culture, microbiology, biotechnology, we are getting so many benefits. Let's talk about the benefit first, then we'll talk about how we can proceed in plant breeding methods. So first of all, with the introduction of plant tissue culture, with the introduction of plant tissue culture, biotechnology and molecular biology, molecular biology, revolutionary changes are done in plant breeding. Conventional plant breeding basically. So in bracket I am writing conventional type. Right, conventional type, so many changes they are being done in this particular direction. Let's talk about what are the benefits we can get from the modern breeding strategies. We'll talk about those or you can say the benefits. We are getting a plant which gives more yield. Now, what does this more yield means? That means we are getting more and more number of the seeds, more and more number of the product. That is defined as a plant with more yield, right? This was the first one. Second, plant products with more nutritive value. With more nutritive value. The third one, we are getting those plants which are resistant to the pest. Resistant to the pest, and next is they are resistant to the bacteria, mycoplasma. So let's talk about those. Plant resistance to bacteria, mycoplasma, fungi, etc. Right? More yield, plant products with more nutritive value, plant resistance to the pest, resistant to the bacteria, mycoplasma and the fungi, and the last but very important is that plants are drought resistant. Now listen to this very carefully. Our ultimate goal is to attain all these type of the characteristics. And we know that these single characteristics is available only in a single type of the variety. Suppose this is available in the single type of variety. Even this is available in the second type of variety. This is available in the third type, and so on, like this they are available. Then what I will do with the help of these different strategies like plant tissue culture, biotechnology, or the introduction of these foreign gene from one species to the another, from one plant to the another, with the help of the transfer mechanism or the use of the vectors, what I can do, I can transfer that particular gene which gives a more yield to my desired plant. I can transfer that particular gene which can provide more nutritive value. Simultaneously, I can provide all these conditions in our plant so that our plant will be more suitable to the different environmental condition, or they can acclimatize to the different environmental condition, right? So this was the major strategies of the plant breeding. Now come at this side. Let's discuss about the different strategies which are opted by the government agency in terms of the plant breeding. Strategies opted by government agencies for plant breeding. Let's discuss all those strategies which are opted by the government. not only by the government, whereas the different agencies, different laboratories who are working on these plant breeding experiments, they are following the same procedure. So the first is called as a collection of the germplasm. So first let's talk about the collection of germplasm. Germplasm or collection of variability. Now, what does this collection of the germplasm or the variability means? Suppose in a variety, suppose for a one particular species, n number of the varieties they are available. So that is called as their germplasm. That means I am collecting all the alleles of the gene. I am collecting all that alleles of the different gene which are there in those species. So that will be called as a germplasm. Now, what will be collected in this? The first of all, the collection will be, I'll be collecting the culturable, which I am culturing those species which are not cultivated as well. I'll collect those species as well. I'll collect those local varieties which are available in the market. I'll take those pure lines which are available at the breeders. So I'll take all those together, and that will be called as my germplasm for that particular variety. So I am focusing on that particular variety. Later on, various modification will be done on those varieties, so that will act as a prerequisite for the plant breeding experiment, because now I am having all the alleles of the gene. I am having the different genes with me which can cause or which is having a major characteristics that can be used in the subsequent generation, and that will cause a benefit in all these terms which I have written over here. Right. So let's talk about what exactly this germplasm is. So germplasm is the collection of all the alleles of a gene, all the alleles of different gene, of all the genes present in a species. So I am having all these which are present in a species. I have collected all those alleles, all those genes. Now I am having all those alternating forms. I am having those forms also that provide resistance. I am having those forms also that provide or that gives a proper yield. So I am having all those kind of things. Now let's talk about what are the different collection that I will be doing when the first prerequisite will be done. Let's talk about those. The first will be the cultivated plant moreover I would rather say cultivated improved plant. So I am using those plants which are being cultivated. Next, I'll take those plants which are not cultivated but they are improved plant but are not cultivated. So these are the total germplasm. Were not cultivated, right? So the third point, let's talk about the third point. The third that I will collect, that is a local population of that particular species, right? Or more precisely, I would rather say desi species which are available in the market. So let's write about the local or desi species, right? Now, next I will collect those which are available at the breeders. So, line produced by breeder. line which is produced by the breeder. Now here line means the defined characteristics is present in that particular population. So that is defined as a line. So we have collected all those things, but we cannot forget a very important, that is you can say those variety which are locally available, or which are more precisely the wild variety of the species, right. So last but not the least, wild species. I'll collect those wild species as well, not all the wild species, but those which are related to the crop. Related to the crops, right. First was the cultivated improved plant. These are all constitute and prerequisite for the plant breeding experiment, right. So they all act as a backbone for plant breeding experiment. So if I'll not be having all these kind of variability, I will not be able to conduct the plant breeding experiments. So this all species, these all characteristics, these all part of the crop which I have collected will act as a prerequisite so that the manipulation can be done in the natural genes, right. So come at this side. All the germplasm collected act as prerequisite for manipulation in gene composition. Right. So first was the collection of the germplasm. I have collected all those germplasm. Now after that, I'll choose the major varieties from that particular germplasm. So first of all, note down this. So students, this was the first part that includes collection of the germplasm or variability. Let's talk about the next part, the second step in this plant breeding experiment. That is evaluation and selection of parents. Now, the second point says the evaluation and the selection of the parents. Now, I have a different alleles, they are present in the gene, I have the different forms of a genes. Now, from that different form of the gene, I have to choose what will be the parents. I have to choose those characters which I want that they should be present in the progeny, and it's a very tedious job because we have to choose those characteristics, and later on I want that characteristics they should be in the pure line, and with the help of their continuous multiplying, I'll get a multiple copies of this. Basically 2 to more than or around 2 of these parents they are being selected. One is chosen for the male and another is chosen for the female in case the plant is a bisexual. In case it is a unisexual, or suppose the unisexual it is, then we cannot define whether we need a plant within a male or the female. Let's talk about the second, evaluation and the selection of the parents. So in this, suitable parents, both male as well as female, is selected right? And they should have desirable characteristics. Desirable characteristics. As they should have a desirable characteristics because the next generation will be having now those characteristics, those characters, right? For example, one of the parent is chosen as a male and second as a female. And other as male, right? In this case, when we need both the parents, right? In those cases, when we do not need both the parents, the species of one of the species, that is important, right? So, in this case, after this, what we'll do? We will multiply these parents. Multiplication of parent plant is done, right? So, this is the next point. That means evaluation has been done, the selection has been done. Now that it's time for multiplying all this. Let's talk about these. Let's talk about the next. Next, I have the parents now. I know that these are the desired characteristics which are present in the parents. Now, what I want is that hybridization experiment should be set on. In this hybridization experiment, it's a very tedious job because I have these two parents, right? In these two parents, I have having an n number of the gene. In this case also, the n number of the genes they are present. The chances that my desired characteristics should come in the progeny is very less. Sometimes it is only 1 in 1000 and 1 in 100. It's very less. So, it's a very tedious job to do this hybridization experiment. In the hybridization experiment, suppose they have both male as well as female is present. Now, in the female, what I will do, suppose I have a female plant. In the female, first of all, I know that I have to do the fertilization experiment. In the fertilization, first of all, I will do the emasculation in the female. Emasculation is a removal of the anther. After this, what's the reason behind removal of this anther is that otherwise, if the anthers they are not removed, there are chances that they may self-fertilize because the stigma is present over there. They have their own anther. The chances that the self-fertilization may occur in these species. So, what I will do, the first of all, removal of the anther as per the bisexual flowers are there. Second strategies that I will do is that after the removal of the anther, I will do the bagging. Right, the bags of the butter paper they are prepared and they used to cover those flowers. Later on, the receptivity is attained. Suppose the receptivity of the flower is attained, then I'll take a collection of our desired anthers. I'll spray those anthers on the flower. After this, the fertilization will occur, right? This is in case of the bisexual flowers. Suppose I have a unisexual flowers. In the unisexual flowers, the emasculation is not necessary. In those cases, what I will do is that the bagging is again done. In this case also, so that the unwanted pollen cannot fertilize those stigma. Otherwise, there are chances that the different variety will be formed. Our desired variety will not be formed. First, in the unisexual also, the bagging is done. Even after the bagging, what I will do is that the spraying of these anthers of the specific anther, the characters which I am looking for will be done on those after removal of the bagging, and later on the rebagging is again done. Right. So, these are the important procedure, that is the bagging and the rebagging, and whole procedure is very tedious because after hybridization, the progeny that will be formed, the fruit that will be formed, the seeds that will be formed, the chances that out of the seed production, mainly only 1 or 2 will get that particular gene. So, it's very difficult to check whether the different type of, you can say the characteristics, the desired characteristics they are present in them or not. Right. So, let us talk about the third, which is called as a hybridization. Hybridization of selected parents. Hybridization of the selected parents. So, hybridization means a mating of the selected parents. Now, let's suppose hybridization in this what will do? Mating of specific parents or desired characteristics parents. They will be done. Now come at this side. Let's discuss about how this mating is done regarding emasculation and bagging. So, after this, let's take an example. Suppose one of the parent is having more protein gene. I am talking about the gene which is present in them. So, the gene between these are the more protein. And let's talk about the second. And in second parent, is having gene for pest resistance. Gene for pest resistance. Suppose these are the two characteristics, they are present in them. After hybridization, there are chances that these two genes will get accumulate in the daughter seeds. After hybridization, the next generation again I am writing might have. It's not necessary that all will have, and choosing from those thousands of the seeds that this is our desired genes, this is our desired characters is a very tedious job. Generation might have, after hybridization the generation might have above 2 characters, right? And the chances of these are 1 in 100 or 1000. Right? Now, after in this experiment, first of all the emasculation is done. The next step in this case is the emasculation. Is done in female flower or the female parent. I am talking about parent. Emasculation means removal of anther, and in this case the female flower, the female parent is a bisexual. So, I am writing the bisexual as well over here, right. Later on what I will do is the bagging. Bagging of flowers is done. Bagging of the flowers is done. After the bagging of the flowers, why this bagging of the flowers is done? To avoid unwanted fertilization. To avoid unwanted fertilization, right? So this was the bagging. Later on, after the attaining of the receptivity, after receptivity of flowers, anthers will be sprayed. Anther will be sprayed and that leads to fertilization. That leads to the fertilization, right? Now, this was in case of the bisexual flowers. I have written over here that is a bisexual. Now, what actually after this the spray, the fertilization will occur, then again we have to rebag those. So, this is a rebagging is again done. Right. Now, the rebagging is done. After the rebagging, suppose let's talk about the unisexual flowers. In unisexual, emasculation is not done. No emasculation. No emasculation is done. And after this, what we do, the bagging is done. Bagging, and the same feature why this bagging is done, to prevent unwanted fertilization, to prevent the entry of the unwanted anther. Similarly, again spray of anthers. Spray of anthers is done, and after the spray of anthers, rebagging again done to ensure the fertilization. To ensure the fertilization, right? So hope you are clear with this, the rebagging and the rebagging, why bagging is done in case of the why bagging is done, why rebagging is done. So this is the second point which states that hybridization of the selected parents, right? Now let's talk about the third, this was the third one. Let's talk about the the next one, fourth one. That means selection and testing of superior recombinant. Now listen to this very carefully. Now we have all those, we have those quality, we have the recombinants that are mixed with the normal one. Now what I will do, what exactly is done in the plant breeding experiment is the selection and the testing of the superior recombinant. Now I look for those which are having the recombinant gene. Recombinant, I have used this word recombinant, I have told you in the previous class as well. Recombinant is that who is having a different gene or the foreign gene inserted into them, that is called as a recombinant gene. Obviously it doesn't have their own gene, rather than they are having some extra gene as well. So that's why I have written over here the recombinant. What I will do, I will select those which are having the recombinant gene in between them. How I can select those? By planting them or by multiplying them. By selfing them or by the homozygosity, by increasing the homozygosity, I can say that okay, this type of characteristics they are present in this type of individuals, these are the desired characteristics. Suppose I am talking about, I have taken the examples that the more protein they are present in them. One of the parent they are having a more protein, other is having the pest resistance. So on that basis I will check whether this particular is having more proteins or not, or they are also resistant to the pest or not. So they are checked for both the characteristics. As they are checked for both the characteristics, the homozygosity, I will increase the homozygosity so that they should have only those genes as well. The alternating form of those genes could be easily eliminated. So this was the fourth, and the fourth one indicate that what we have to do, increase homozygosity. I have to increase homozygosity. Why? because I don't want any alternative alleles. So I'm writing over here no alternating allele. No alternating allele should be present in them, right? So this was the fourth, which is called as a selection and testing of the superior recombinant, right? So students, note down. So students, let's talk about the next step in the plant breeding, that is the testing, release and commercialization. The last one. The fifth one is the testing, release and commercialization. Now, what does this testing, release and the commercialization mean? Now, all those seeds which are having those recombinant gene, all those recombinant seeds, they are being sent to the laboratory. Basically, in New Delhi, we have ICAR, that is Indian Council of Agriculture Research, New Delhi. It is present over there, and they used to evaluate whether such type of genes they are present in them or not by growing in the area, growing in the large area. By growing those seeds in the large area in the natural climatic conditions, later on they are again sent to the different region. There are 3 agroclimatic regions. They are sent to the 3 agroclimatic regions, because we want to know whether they can adapt to the different environmental conditions or not. After this particular testing in the different environmental condition, they are being sent to the various, you can say, so the seed agency, national seed agency, they are sent over there, and later on they are commercialized, right? Let's talk about the testing and the release and commercialization. In this part, seeds, I am talking about the recombinant one, sent to ICAR. Indian Council of Agricultural Research. New Delhi, right? New Delhi. In this, the testing is done on the seeds. First of all, they are grown on agricultural land later on, and in agricultural land they are given proper fertilizers, with proper fertilizers Later on, these seeds are sent to 3 agro-environmental condition, agroclimatic zones. So, sent to 3 agroclimatic zones. 3 agroclimatic zone, whether they can grow in this particular environmental conditions or not, because we know that we have a 3 agroclimatic conditions, 3 agroclimatic zones we have. On these particular zones, whether our seeds they grow or not. So that is the biggest achievement for those if they grow on these regions, and later on this is grown for 3 generations. If they are successful in these 3 agroclimatic zones, they are sent to National Seed Corporation, NSC. They check whether they are suitable or they are more improved as compared to those varieties which are available in the market or not. Later on, they are commercialized and they are sold. Commercialized and they are sold in the market. Right. So these are the 5 basic procedures, these are the 5 basic points by which a selection in the germplasm to the hybridization and later on the commercialization of the seeds is being done. Right. So this was all about the plant breeding. So thank you so much students for watching this. We'll meet in the next lecture.